Okay, right stain. Right stain is part of something called the Romanowski stain. It's a very, very broad category of stain that uses polychromatic type dyes. So you're going to have cells that actually stain in the whole rainbow of colors. Okay. Some of you probably had microbiology, so you had gram stain before. Yeah, so things either turn blue or things turn red, one or the other, right? Or if you use something like India ink, basically you have a dark area or light area. But with the right stain, Romanowski stain, you're going to have a whole spectrum of cells. Okay? And it's based on the acidity of the cell components. Okay? We have parts of the cell that like acidic dye, and other parts of the cell that like basic dye. And the acidic dye we use is called the eosin, or acidophilic components. It will stain acidophilic components. And methylene blue is the other part of the dye that we use that will stain the basophilic, or base-loving components of the cell. Okay. So sometimes you see a pathology report or histology report, you might hear terms like, this cell has basophilic inclusion. That means that part of the cell stains blue color with methylene blue. Or you might say eosinophilic component or acidophilic component. That means that part of the stain that stains orange color. Okay? But the combination makes it possible for the cells to have all different colors, from yellow to green to brown to just about any color. Okay? So it's really, really helpful, and it's the most commonly used stain we use in the hematology lab. Something related to the right stain is called the Leishman stain. Maybe you've heard of that. They use that in the parasitology microbiology courses a lot because they tend to stain the parasites a little bit better than the right stain. But in hematology, we basically use the right stain. Pretty much the same thing. Okay? We're not going to spend a whole lot of time with smear making and so forth, but essentially what we do is have a technologist or a lab assistant who draw the blood samples for us to make a thin blood smear. This is a thin blood smear being made on 2 microscope slides. Is it better if we turn the light off? We don't want it too dark so you fall asleep. But anyway, this is making a thin blood smear using 2 microscope slides. And you can see it takes a little bit of practice, but this area here is the thin area and this is the thick area. And what we're interested in is about 2 fields, microscopic fields, in from the most thinnest part of the smear. And that's where the cells are just barely touching each other and they're not overlapping. Okay. Then in that area we can actually examine the morphology a lot better. Okay. We call that the wedge technique, and there are a couple of different techniques we can use to make smears, but this is the one that we use mostly. Okay. So these are just examples of thin smears, and you can see this is called a feather edge, and a couple of fields in is the area where we want to take a look at under the microscope. And you can see that some slides have red color staining primarily, and some are kind of bluish color, and that tells you that depending on the specimen and depending on the condition of the lab, you can have the cell stain more blue color or more red color. It really depends on the pH. Yeah. So in the old days, like before my time actually, sometimes we will have a chemistry section right next to a hematology section in the lab, and the acid fume from chemistry section will migrate into the hematology staining area and it will affect the right stain. Okay, so we have to be careful about the fume inside the lab as well. But today everything is pretty much automated, so we don't have to worry about that. Okay, any questions so far? All right. In the hematology lab, one of the most commonly performed manual technique is called the differential count. Of course, we have the instruments to count the red cells and white cells for us, but we also have to look at these cells under the microscope and differentiate them into different white cell types or identify abnormalities in terms of morphology, red cells and white cell morphology. So to do that, we do something called differential count, or simply diff. Okay, so that involves leukocyte differentials, and we also estimate the number of platelets in circulation from the slides. and we examine the red cell morphology. Erythrocyte is a scientific name for red cells, and leukocyte is a white cell, yeah? And this is just enhanced scanning EM showing you the different types of cells you might see in circulation. Red cells, human red cells are those round red ones, and they have no nuclei. And we'll come back to red cells a little bit later on. But normal human red cells in circulation have no nuclei. Okay? So it's pretty, bland looking thing. And then we have these white little round things. These are probably lymphocytes, white cells. They look a little smaller, but it's because of the three-dimensional structure. Once they're on a microscope slide, they flatten out a little bit, so they look a little bit bigger than that. And we have platelets in circulation. So what we're going to look at are not the three-dimensional pictures, but two-dimensional pictures. But in your mind, try to remember that the cells you see under the microscope are really three-dimensional. And there are evidence. There are places where you can see that the cells have thickness. And that's kind of important, especially with red cells. So we'll come back to that. In order to identify the cells, to name the cells, there are several things you need to look at. But remember, not one of these key features are going to actually diagnose the cell. You're going to have to look at the whole thing, every little key features, and try to think of the pattern. Okay? So, in general, it's true that the lymphocytes are smaller than neutrophils. Okay? Those are different types of white cells. But it's not always true. Okay? And it's also important for you to recognize size anyway, because certain types of anemia are characterized by small red cells, and other types of anemia might be large red cells. And we'll come back to that. Nucleus shape is actually pretty important. If you see nucleus in a cell, some cells, like lymphocytes, characteristically will have round nucleus, whereas neutrophils will have very complex lobulated nucleus. And that's the kind of feature that we look at. Chromatins, the DNA strands that you can actually see inside the nucleus, is actually very, very important, especially if you're looking at precursor cells. We don't see a lot of precursors in circulation, but if you happen to tap into bone marrow and look at the cells right at the origin of the red cells of white cell production, you can actually see the precursor cells. You can actually determine how young the cell is by looking at the chromatin feature. Okay? But we're not going to go too much into that. We also look at the cytoplasm of the cells and see if there's any inclusions in it. Large inclusions, small inclusions, what color are they, and that determines what kind of cells are there. And of course color. Okay. Again, bright stain stains different parts of the cell according to pH. It's a polychrome stain, so it's kind of important. But did you know that a lot of pathologists are colorblind? So they don't really rely that much on color, and you can actually do the work without color. Okay. In fact, if you happen to have one of these computer atlas, try to turn off the color. You should be able to still identify the cell. Okay. So color is neat. I mean, it's very helpful, but it's not that essential. Okay? In fact, depending on the brand of bright stain you buy from companies, sometimes they don't stain the same way. So if you look at your textbook, and they'll tell you eosinophil granules are a nice bright orange color. Well, in real life, it's more like brown color. Okay, so don't rely on color that much, but it is helpful. Okay, so let's start with leukocytes. We're going to go through leukocytes. We're going to do a little bit of platelets, and then come back and do red cells. And today I'm going to show you essentially all the normal leukocytes in circulation. I'm not going to show you in leukemia cells or cells with infections, not that much anyway, okay? But all the basics. You need to have the basics first before we can go into the abnormals. Okay, so with the differential count, one of the most important things is for you to be able to differentiate the different types of white blood cells in circulation. And there are 5. Eosinophils, basophils, neutrophils, lymphocytes, and monocytes. That's pretty simple, right? That's 1 out of 5. So if you have multiple choice, well, let's not talk about that. But we're going to make it easier for you. Once you identify a white cell, which is actually pretty easy to do because white cells are the ones with nuclei. Red cells have no nuclei, remember? So once you identify a white cell, you can determine if you're looking at a granulocyte or non-granulocyte. And that is according to the presence or absence of specific granules in the cytoplasm. Specific granules. So the term specific is a key word. Okay. Non-granulocytes can have things in the cytoplasm, but we don't call those specific granules. Okay. Now, once you have a granulocyte, you only have 3 choices. That's 33% chance, so even better on the multiple choice. Eosinophil, basophil, neutrophil. If you decide that you're looking at non-granulocyte, that's even better, 50-50 chance. Lymphocyte or monocyte. Okay, so let's start with the granulocytes. And I'm going to start with eosinophil because it's a little bit easier to look at and give you examples of the features that we're talking about. Although you don't really see that many eosinophils in circulation normally. Okay, characteristic, all granulocytes. All white cells have nuclei. So these purple things in here are nuclei. And in the cytoplasm you have these little things that look like, in this case, look like jelly beans. Those are the granules in the cytoplasm. So all granulocytes are essentially about 14 to 16 microns diameter on the microscope slide. Okay. And it's kind of flat looking. And then they'll all have irregular looking nuclei. And in the case of eosinophil, it's a single nucleus, but it's separated into 2 separate lobes. It's connected by a little thin filament, which is a fused nuclear membrane. So you have 2 lobes connected by a filament. Okay. And eosinophils are characterized by presence of orange-brown large granules in the cytoplasm. These granules are about 2 microns diameter. They're generally uniform in appearance and size, so that's why I like to call them like orange jelly beans. Okay. In normal adult blood circulation, there's usually less than 2% in circulation. 2% of the white cells will be eosinophil. So it's pretty rare. But you will see increase in eosinophil count associated with things like allergic reaction, hay fever. Springtime is usually that kind of season for, especially on mainland. Yeah. So you're going to see people with more of these reactions. Patients with asthma, they might have increased eosinophils. And parasite infections like roundworm infections or even tapeworm infections, you can have increase in the eosinophil count. One of the cases that we actually use in the lab, in our teaching lab, is a case where a patient actually has something like 65% eosinophil. That's a huge, huge increase. Very, very obvious. And this was due to roundworm infection. Any questions on eosinophil? Pretty straightforward so far, right? By the way, we're going to have a little quiz later on. Okay, next granulocyte I'm going to show you also has a very obvious feature of lots and lots of granules in the cytoplasm. So that's why I use this for the next example. But this one is even more rare to find in circulation normally. These are called the basophil because these granules like the basophilic dye, methylene blue. So that's why these granules stain very, very dark. Very, very dark. Your textbooks will tell you the granules are dark purple. Usually they say dark purple, but it's more like almost black. And they look kind of like charcoal. The granules are not always uniform in size. They're kind of random size. Sometimes they even look like a little irregular thing, like broken up charcoal thing. So, but this case here, this example here, you have the granules covering the almost entire cell, so you can hardly see the nucleus. But some basophils may have just a few granules, so you can actually see the nuclei. Okay? So this is not always the case with all the basophils. But if you can see the nucleus, it'll be probably bilobe, maybe 3 lobes at most, sort of like eosinophil. Okay. Size is about the same, about 14-15 microns diameter, and less than 1% in circulation. When we do differential count on a screening basis, we basically look at 100 consecutive white blood cells and try to differentiate them into different white cell types, right? So if we say less than 1%, that means a lot of times we don't see any basophils when we do normal screens. Okay? But these cells are found in tissues like skin, in the lungs, and other areas. So that's where they do most of the job, but not so much in circulation. Yeah. So for example, when you scratch yourself and you get a red mark, you actually cause the content of the granules from basophils to come out from the skin. So that's why you have a little capillary dilate, and you're going to have a red mark. Okay? But in circulation, we don't see a lot, except in like allergic reaction, asthma, hay fever, and a very, very important disease called CML, chronic myelocytic leukemia. Chronic myelocytic leukemia. Okay. This is finding an increase in basophil count is one of the hallmark for diagnosis of CML. So that's how important that is.
Sorry, what was the M acronym for?
M is myelocytic.
Myelocytic?
Myelo means bone marrow, so it's a white cell leukemia from the bone marrow, but it's a chronic disease. Okay, it's a type of leukemia, and some of these patients actually turn into acute leukemia later on, and if they're not treated, it could be a fatal thing. Okay. Any other questions? Okay, just a couple of other examples, different examples of, actually they're both basophils. Okay, on the one on the left is more obvious. Okay, you have dark inclusions in the cytoplasm, but this is not as filled with inclusions. So you can actually see the nuclei, the purple nuclei inside. It's a bilobe nucleus. It's a single nucleus, but it's separated into 2 lobes. So it's still one nucleus cell. This one here, on the right side, the granules look more lighter purplish color, yeah? But that's because of the staining process. You can see the red cell color is different. This red cell and this red cell, the color is a little bit different. So the entire smear is more acidic rather than basic. So that's why everything turns a little bit more to the reddish, orangish, pinkish color. But this is also another example of a basophil. So be careful when you rely too much on color. And finally, the granulocyte neutrophil. Okay, this is the most abundant form of granulocyte you see in normal adult circulation. Okay, 50%, 60%, up to maybe about 70% of the cells will be segmented neutrophils. And there's so many of them that you can actually find some neutrophils that are not as mature, so they're not as segmented. So for example, this one here has a nucleus that's segmented into a couple of lobes here, and it's connected by a nuclear membrane over there. But this particular ropey thing here still has the nuclear material inside, so we wouldn't call that a filament. Filament would be a very, very thin thread-like. But so this is not quite 100% mature yet, and these cells are not mature at all, so you don't see any filament at all, right? So these cells are called a band form, and something like this is called a segmented form. So we differentiate segmented neutrophils from band neutrophils. They're about 14, 15, 16 microns in diameter, just like any other granulocytes. And the granules are not as large, they're pretty small, and they don't stain as well as basophils or eosinophils. So that's why we call them neutrophil. Okay? But they do stain light purplish color. You can see the small granules in the cytoplasm. The chromatin pattern is very obvious, very dark, thick chromatin pattern. We call that coarse pattern. So you have a thick, dark area, and then you also have a very obvious light area. So parachromatin, euchromatin, differentiation is very obvious. And you see increase in this particular cell line, especially with an increase in band count associated with bacterial infection. Bacterial infection. Okay. So next time, yeah, these two are called the band. Okay, and this one is closer to segmented form, but strictly speaking, this is not a segmented neutrophil just yet, because this line here is just a little bit too thick. Maybe, what, I don't know. Okay, here. This, you can see this filament is a lot thinner, yeah? More thread-like. This one is pretty thin. Okay. So next time you see a TV show like House or something, and somebody's talking about high white cell count with a shift to the left, high band count, things like that, they're referring to bacterial infection in the patient. Okay. High white count is associated with some kind of infection anyway, right? And shift to the left means you have increase in a younger population of that particular cell line. Okay, so when you draw a curve, the frequency distribution curve of a cell, the curve shifts to the left towards a younger population. So that's what it means by shift to the left. Okay, so more bands, that means younger cells. That's a shift to the left. That means bacterial infection. Okay, more examples of a segmented neutrophil. On the top, this is a very obvious segmented neutrophil, pretty healthy looking. This one also healthy looking, and you can see that neutrophils tend to be a little bit more lobulated than eosinophils. They may have 3 lobes, may have 4 lobes, generally not 5 lobes. Okay, 5 or more sometimes indicates that the patient has some kind of disease process. This one here is probably a band. It may be a mono, but I'd say you can see that this is a nucleus is not lobulated, so this is not a segmented form. But this one is a segmented form. This one is a segmented form. Okay. Any questions so far? Okay. So granulocytes. Recap. We got eosinophils. If the granules stain with eosin. So more like orange, purplish, orange pinkish inclusions, and basophils if the granules stain with methylene blue, and you get black, dark purple inclusions, and neutrophils if the granules don't stain that well, but you can see them, the small granules in the cytoplasm, and it's most abundant form, neutrophils. Okay. Now we go to non-granulocytes. And there are only 2, lymphocytes or monocytes. And we'll start with the lymphocyte. Lymphocytes are relatively small, but they vary in size. Okay? Now remember, granulocytes, I told you, are about 15, 16 micron diameter. Lymphocytes can vary from small, mature lymphocyte like this one here on the right side. They're about maybe 10, 12 microns diameter, to the one on the left side, which is also a normal lymphocyte, is about 20, 25 microns diameter. So it varies. But generally speaking, lymphocytes should have round or oval nuclei. They never have lobulated nuclei. They don't have convoluted, twisted nuclei. They should not even have creases in the nucleus. No folding, no creases, no indentations, deep indentations in nuclei. In fact, creases and deep indentations in nuclei in lymphocyte is considered to be very abnormal. We may be looking at leukemia or lymphoma, so that's one of those clues. So normally round or oval nucleus. The chromatin pattern tends to be less defined, so they're not as coarsely clumped like granulocytes. So we call them smudged chromatin pattern. Cytoplasm can be blue color, like this one here. We call it basophilic. Okay. One of the functions of lymphocyte, remember, is to make antibodies, right? Remember that? And antibodies are protein, yeah? So in order to make proteins, what else? What do you need? you need the ribosomes, right? Yeah. And ribosomes like methylene blue. So if the cell is actively making a lot of proteins, the cytoplasm tends to be very blue. Basophilic cytoplasm. Okay? But some cells, lymphocytes may not have a lot of ribosomes, so they don't stain that blue. And you can see from this side, there are some purplish inclusions in the cytoplasm. So these cells can have things inside the cytoplasm, but we don't call them specific granules. Okay? Specific granules are only those we consider to be basophilic, eosinophilic, or neutrophilic. These other ones, we have other names for it, but we don't consider these the same as granulocyte granules. Lymphocyte count is generally only about 25%, 30%, 35% at most in adults. In children, preteens, their lymphocyte count tends to be a little bit higher because their lymphoid organs are still growing, they're developing, so they tend to put out more lymphocytes in circulation. So it could be maybe 40%, 50% in circulation for kids. But if you begin to see 50%, 60% in adults our age, then we consider viral infection, virus. Okay? Now remember, with the neutrophils, I said high neutrophil count is associated with bacterial infection. Now high lymphocyte count is associated with virus infection. So a little bit of difference there. Okay? If you see 60%, 70%, 80% lymphocytes in somebody older, like my age and older, then we start thinking about leukemia. So that's also not a really good thing. Okay. Any questions so far on lymphocyte? Pretty simple, right? Okay. Now in your textbook, you might also see that these purplish inclusions in the lymphocytes, you see more of those with T cells, which is probably true, and we tend to see more of those in T cells. But morphologically, T cells and B cells, you can't really tell the difference. Okay, so don't rely on these inclusions to try to differentiate T cells and B cells. There are other ways. There are more immunologic ways you can use to differentiate the two, so don't rely on that. The other non-granulocyte is the monocyte. Okay. Monocytes are relatively large. On the top here, this is a monocyte, and down here is a segmented neutrophil, just for comparison. So monocytes are bigger. They're about 25 microns diameter, very large. The nucleus tends to be very irregular. It could be U-shaped, horse-shaped, twisted, convoluted, could be lobulated, all different kinds of shapes. Cytoplasm is generally gray color, sometimes bluish, but usually more like a gray color. Sometimes they're described as ground glass appearance, like etched glass appearance. You can have vacuoles and you can have inclusions because monocytes are phagocytic cells, and one of its jobs is to identify old cells, dying cells, or invading organisms and eat them up. So they're like garbage collector. So you can have lots of vacuoles in the cytoplasm of monocytes, and you can have inclusions. Chromatin pattern, if you can actually see them under a microscope, do they can just use the microscope here? Mm-hmm. Oh, okay. If you see monocytes on the microscope, the chromatin pattern tends to be a lot more delicate looking. We call it streaky, linear chromatin arrangements, very delicate looking. The monocyte in circulation, normal adults, is only about 3% to 7%, so not very many. But you can have increase in monocyte circulation with certain bacterial infections, chronic bacterial infections like tuberculosis. You can also have increase in bacterial infections after the infection is gone, like a recovery phase of infection. You can also have monocytosis with certain types of leukemia, but that's pretty rare. Okay. Any questions so far? So white cells, granulocytes and non-granulocytes, right? Granulocytes, we've got three: eosinophils, or basophils, or neutrophils. And neutrophils, we have so many of them, so we can differentiate them into either band stage or segmented stage. Okay. Non-granulocytes, you only got two: lymphocytes or monocytes. No problem, right? Okay. Just a little bit on platelets. They're also called thrombocytes, but they're really not cells. They're actually fragments of the cytoplasm of this huge cell in the bone marrow called the megakaryocyte. Megakaryocytes can be like 100 microns diameter. It's a huge cell. It's very easy to spot even under low power. Okay? But this particular cell, which comes from the same stem cell as white cells and red cells, but they develop into this large cell because it keeps dividing without actually physically separating. We call it endomitosis. So it gets bigger and bigger and bigger. And when it's finally mature, the cytoplasm matures into little bits and pieces of platelets, like these little round things. They're about 2 microns diameter round things. And they kind of flake off and gets into circulation. And the rest of the cell will be recycled. Okay, so what we see in circulation are bits and pieces of cytoplasm. We call them platelets. They should have granules. Okay, on the right stain, you should be able to find small, tiny purplish granules inside the platelets, and that's very essential. They contain materials needed for platelets to function, as in plugging up damaged blood vessels or activating the clotting factors. Okay, make sure that the clot actually begins when it's supposed to. Okay, and without that, the platelets won't work, so the patient will continue to bleed. We can do an estimate of number of platelets in circulation. We can do the estimate for white cells too, but that's not as accurate, so we don't do that too well. But with platelets, we can do a better job with estimating. If you look at the oil immersion field, on the average, you should see anywhere between 8 to 20 platelets per oil immersion field. So take a look at several fields, and you can average them out. And if the average number happens to be within 8 to 20, that roughly corresponds to about 150,000 to 400,000 platelets per microliter, which is within the reference range. Reference range, normal range, pretty much the same thing. In the lab, we use the term reference range. So we don't want to say something's normal, something's abnormal, right? We say who's normal, right? So reference range of platelets, 150,000 to 400,000. That sounds like a lot, right? 1 microliter is like, it's a tiny fraction, like 1/100th of a drop. Okay? And that small amount of blood, there are 150,000, 400,000. In the same volume of blood, there are about 5 to 10,000 white cells in circulation. About how many red cells do you think we see in 1 microliter? Any guess? 100,000? Actually,
I just said double the number.
double number is actually more than that. It's more like 6 million. Yeah. 6 million red cells in one tiny little fraction of a drop. And in old days, actually the old days, my days, we used to have to learn how to count those cells on the microscope manually, click, click, click. Okay, of course, we don't do a million cells, so we dilute the sample, so we only count maybe a few hundred. But still, after a day of doing that, you can cross eyes. Anyway, so platelets in circulation, 150,000 to 400,000. So we can do an estimate. Okay, and that's part of the differential pattern. Okay, now comes the fun part, the red blood cells. Okay, they come from the same stem cell as white cells and platelets, but just before it's ready to be released into circulation, the maturing red cells loses its nucleus in the bone marrow. And it stays in the bone marrow without the nucleus for a couple of days. Then it gets out in circulation and then mature for a day or so. Okay? And then once in circulation, under normal circumstances, it will circulate for about 120 days. and then gets recycled. Okay, that's a normal lifespan. They're generally uniform in size and shape. They're biconcave disc of about 7.5 microns diameter. They're pretty uniform normally. If they're not uniform, we got disease state. Biconcave means you got this indentation from the top, and you also have indentation from the bottom. Okay. Now some of these pictures, they may kind of look like Cheerio or donuts, but there's no hole through the middle. Okay. It's more like malasada, and you flatten the middle, and then you get this biconcave shape. Getting hungry?
Yeah.
Okay. But remember that we're looking at three-dimensional cells, so you can see that outside of the cell is thicker than the inside of the cell, right? So if you look at these cells under the microscope in two-dimensional fashion, the outside is darker color than the middle part. Okay? In fact, the middle part is going to be very, very pale looking, and that pale area is roughly about 1/3 of the cell's diameter, and that would be a normal red cell coloration. So central pallor, approximately 1/3 of the cell diameter. Okay. Now, inside the red cells are all kinds of things. There's some protein and so forth, and very important protein is the hemoglobin. Yeah, but there is no inclusion, no nucleus, no chromosome, no granules. It should be nice and smooth. And this is under a medium low power field. You can see a field of red blood cells, and each of these red cells have central pale area. Okay, and central pale area should be about 1/3 of the cell's diameter. And this is a normal looking red cells. Okay, and this is a kind of nice area to look at because you can see red cells are just occasionally touching each other, but they're not excessively overlapping. And so this is a good area to do the morphology studies. Okay, just a reminder that we're looking at three-dimensional red blood cells, but not two-dimensional flat thing. But if you find variations in the size or the shape, then we need to report that. We need to recognize that because it's a good, important clue as to the kind of disease a patient might have. By morphology, you can steer the diagnosis into the type of anemia we're looking at. Okay? If a red cell size on the average is less than 7.5 microns diameter, we call them microcytes. If it's larger than 7.5 microns, then we say macrocytes. That's pretty obvious, yeah? Now, without the micrometer, it's kind of hard to determine how big the cells are. But after you look at a bunch of examples, then you can tell if you're looking at small cells or large cells. Yeah. But I'll show you in a little bit how you might actually determine an average cell size. But these cells are small, and you can see the central pale area for a lot of these cells are greater than 1/3. So they're also more pale in color. So small cells, microcytes, and pale color cells, they're associated with iron deficiency. Iron deficiency. Okay, here on this side we have big cells. This is a segmented neutrophil about 15 microns diameter. So these cells are bigger than 7.5. This is about 7.5 normal looking red cells, but these cells are large cells. So these are macrocytes. Could be associated with liver disease, and it could be associated with things like vitamin B12 deficiency, folic acid deficiency. So certain types of anemia can give you large cells like that. Oh, these cells on the average is about 6 microns diameter. But it can get pretty small. It can get maybe down to about 4 microns or maybe even less than that.
Yes, less than 7.5.
Less than 7.5.
More than 7.5.
Macrocytes, yeah. Macrocytes would be big, like these guys, about 10 microns. Yeah. So variation in size is called anisocytosis. Anisocytosis. But you can also find variations in shape. Normally they're biconcave disc, round, okay, with a central pale area. but sometimes they are not biconcave shaped. They're not even round. And when we find those things, we say poikilocytosis. Poikilocytosis. Okay, here we have a cell that is round, and it has a dark area on the outside, and then we have a light area, and then we have a dark area again. So it looks kind of like a bullseye on a target, right? So we call these target cells. Okay, and they tend to have a excess amount of membrane for the content, so that's why it actually looks like a little hat in circulation. But when it sits in a flat surface like the last slide, the center part bulges up, so that's why you have this little dark area in the middle. They're called target cells, and they're commonly associated with hemoglobin disorders like sickle cell disease or thalassemia. Target cells are a very, very important clue as to diagnosing the genetic disorders involving hemoglobin. Okay. On the right side, we have examples of small cells again, but with large central pale area. So if the cells, red cells, are pale in color with large central pale area, we call that hypochromic. Okay, so microcytic hypochromic is almost automatically iron deficiency, but not always. Okay, nothing is all that clear cut. Now this slide I want to show you because here we have a normal small lymphocyte. Okay, round nucleus, a little bit of blue cytoplasm. If you find a normal, mature, small lymphocyte, the size of nucleus, diameter of nucleus, is about 8 microns diameter. So it's a good size comparison to a normal red cell. Yeah? And you can see if this is about 8 microns diameter lymphocyte nucleus, these cells are much smaller, right? So obviously we have microcytic and also hypochromic cells. And everybody's familiar with sickle cells. Okay, sickle cells have abnormal hemoglobin, sickle hemoglobin, which makes that particular hemoglobin more sticky, I guess, so they kind of line up when oxygen is removed. And by doing that, these cells deform into the sickle-like shape. And what happens is they get stuck in the capillaries and cause very painful sickle crisis, and it can actually cause death. So if you have a patient with homozygous sickle cells, then you can actually find those sickled cells. But hemoglobin S is not the only hemoglobin that will sickle or deform the cells. So having the morphology changes like this does not necessarily make a final diagnosis of hemoglobin S sickle disease. Okay? You can have hemoglobin C disease, you can have SC disease, you can have a whole bunch of other hemoglobins that can actually deform the cells. Okay? But it is a hemoglobin disorder, so you have target cells in the background. Occasionally you find fragments of red cells. We call these schistocytes. Schistocytes. It could be fragmentation mechanically due to maybe this clotting that's happening inside the blood vessel. We call that the fibrin strand formation. And red cells, when they pass through those fibrin strands, they get sheared off and they get broken up, and you end up with little fragments. Or maybe there's some mechanical devices like a defective heart valve or something that can actually damage the red cells. That can give you schistocytes. Okay. And we use common terms. Okay, so you can see cells like that. It looks like a teardrop, so we call them teardrop cells. Sometimes they look like pear-shaped, so we call them pear-shaped cells. Sometimes this teardrop, this section here gets a little longer, so some people call them hand mirror cells. We have all kinds of different names. But this is also a type of poikilocytosis, and it could be associated with certain diseases like myelofibrosis, but it's not as diagnostic, but it's something that we pay attention to. Okay. And on the right side we have red cells that look real dark, but it's really not dark because it has more hemoglobin, but it's dark because the central pale area is not there anymore. A central pale area means you have biconcave shape, yeah? The flattened monocyte. Now, if you don't have a central pale area, that means cell is more spherical. Okay? So we call these spherocytes. Okay? Although they're dark, we don't say hyperchromic. We just say spherocytic. Spherocytes. Okay, and other pretty rare conditions. Elliptocytes. Red cells should be nice and round, but sometimes by genetic defect, the cell membrane proteins are arranged in a different way, so you end up with these elongated red cells we call elliptocytes. Elliptocytes. Okay, here's a small normal lymphocyte size comparison, so they look a little bit smaller too. Okay. Now we're concentrating on normal human adult red cell morphology here. If you look at cells from other animals, they're not always like human. For example, camel blood is normally elliptocytic. So, here we're only talking about humans, yeah? On the right side, we have cells, round, and it has a central pale area, but the central pale area is not round, but well, I guess you have to stretch your imagination. Does this kind of look like a mouth with a big fat lips? We call these stomatocytes, meaning it looks like a mouth. Okay? The central pale area looks a little bit like a slit. So it makes this whole cell look a little bit like a mouth. So we call it stomatocyte. And it's also associated with defect in the cell membrane protein structures. One of the conditions associated with that has to do with Rh antigens. And later on, Sherry will be here and she'll talk about the Rh blood group. But Rh antigen is one of those proteins that's associated with the red cell membrane. And there are very rare patients who have no Rh antigens at all. And we're not even talking about Rh negatives. These patients have not even the basic structure of Rh proteins. So then the whole red cell membrane would be a little bit different, yeah? And that causes the red cells to deform into this shape. We call it Rh null syndrome. Very, very rare condition. Okay? But probably you're not going to see one of those. So it's one of those rare, almost trivial thing for you. Okay. Now a few other things, color changes and inclusions. This cell here, this is a red cell. And these are also red cells. These are pretty normal looking red cells. But this one here, does it look a little bit bigger? And does it also look a little bit more purple color? Yeah? Gonna have to look at it real closely. But it's purple because it's taking up the methylene blue a little bit more than the other red cells. And remember what methylene blue like? Basically, yeah, things like RNA, ribosomes, stuff like that, right? So this cell, this particular red cell, is a cell that just got out of the bone marrow. So it's still active in making hemoglobin. And hemoglobin is a protein, so it's got a lot of ribosomes, right? So when you begin to see a lot of these purplish large cells, we call it polychromasia, we tend to think active bone marrow red cell production. Active bone marrow red cell production. Maybe this is in response to anemia or hemolysis or bleeding. And on this side, we have these purplish inclusions. They're about 1 micron diameter, dark purple, usually round inclusion. And that color looks just like a nuclear color, right? Nucleus. In fact, these are pieces of chromosomes. Okay, chromosomes that somehow got left behind when cells divided early on. It didn't get incorporated into the nuclear membrane. So we call these Howell-Jolly bodies. Howell-Jolly bodies. Okay, and presence of that, many of these can indicate certain disease conditions, so we have to be aware of that. So remember, polychromasia is RNA. Howell-Jolly bodies are DNA. Okay, more. Yeah.
That's actually, is that, when you say Rh antigen, stomatocyte, is that with Rh antigen, without Rh antigen?
Oh, that example is a very rare example. 1 in a million patients or something like that have no Rh antigen, and these patients can show stomatocytes. But it's not the only condition. So morphology is great. It gives you clues, but it's not that diagnostic. Okay, cavit ring. Okay, this nice purple ring. It could be a full ring like that. It could be partial ring, a little arc. It could be figure 8 ring. But we tend to think of the cavit ring as a remnant of spindle fiber from cell division. So this is kind of related to hollow jolly body in a sense that maybe there was some problems with cell division, so you ended up with these cavit ring. Normally after cell divides, all these things should dissolve, but if you have these persisting to this stage, then we tend to report it. On this side is a little bit more common. These are called basophilic stippling. Basophilic because these little things, inclusions, like the methylene blue, so it tends to stain kind of purplish colored. And you have a lot of these. Sometimes they're very coarse stipples, like the one down below, here, or small fine stipples like that. They tend to be associated with heavy metal poisoning, like lead poisoning. And it could be a very important clue. Again, it's not very diagnostic, but it could be a very important clue. So, patients who may live in an area where water is contaminated with lead, there was some scare about that on the Big Island a few years back, yeah? So all the kids were tested for lead levels, and if they were to look at the red cells, maybe they were looking for basically stiffness like that. Do you watch CSI? Some of you do. Many years ago, there was one of the episodes where this gambler died on the gambling table, and after they went through the whole investigation, they figured that he died of poisoning from lead because he liked to eat a lot of chocolate, and the chocolate came from a country that was still using a lot of lead as gasoline. So that's how he accumulated lead in the system. And they were describing about basophilic stippling in the CSI episode, so I thought it was neat, except the picture was wrong. But anyway, that's all right. They tried. Okay, rouleau formation. In some conditions, the blood can be really, really thick, so these red cells can stack up like poker chips or something. Okay, that's pretty rare, but it is also another indication of rouleau formation. And then you can have parasites and microorganisms that infect the blood, and you can actually find them sometimes. Now, on the right side, either 1 or 2 is a malaria. Guess which one it is. How many say malaria must be number 1? How many say malaria must be number 2? Okay, and how many say both? Okay, and how many say neither of them? Can't be, right? You're very good at taking multiple choice exam. Actually, it's number 1. Okay, number 1 shows a very small ring-formed malaria that's in the red cell. This part of the life cycle, the organism is actually in the red cell, and it has a little dark purple area right here. Okay, so this is a ring-formed malaria. This is a platelet sitting on top of the red cell. So you can see around the platelet is a halo, white area. So you can see it's a platelet sitting on top and then pushing down on the red cell. So that's why you have the halo. But malaria can be found in red cells. And do we see malaria here in Hawaii? You know? Actually, we can, because people go to malaria-infested places and come back with it sometimes, especially soldiers. They come back with it sometimes, so we can find them. At Tripler, sometimes they find more often than the rest of the hospitals, but we see that. But we don't have the right vector here to transmit malaria from people to people, so we don't see the actual infection here, active infection here. Yeah? But apparently in Hawaii we have bird malaria. So if some of you are going to be vets, for example, maybe you'd be involved with looking at bird malaria in Hawaii. Okay, just a few artifacts. This is an example of an area that's too thick and also very alkaline. So everything is dark purple, blue color, and everything is stacked on top of each other, and you can't really tell the morphology that well. And if you also go into a very thin area, everything is all flat, and they look like a huge spherocyte, because not all these red cells have central pale area. Even the white cells look real flat. And also, if it's too acidic, then everything turns pinkish color. So just an artifact. And more artifacts. We have these little precipitates. These are precipitates from the stain. They kind of look like platelets, but they're not. In this field, this here is a platelet. This is a platelet. This is not a platelet. Okay, so you're going to have to be careful about that. Here we have clumps of platelets. Okay, this kind of sample is not acceptable in hematology because it alters the distribution of cells. So we're going to have to go back and then redraw the patient so we can get a sample that's not clumpy like that. On the other hand, it tells you that patient's platelets are working. So in a sense, it's a kind of platelet function test. But there are other ways to do that. So. And of course, we don't want half a cells, yeah? These smears are pretty fragile. So unless they're cover slip, be careful when you wipe off your oil if you use oil immersion lens. Okay, any question? Where's time? Where's time? Okay, what do you say this cell is? Would you say it's a granulocyte or non-granulocyte? It looks like a granulocyte. Then you got three choices, right? Eosinophils, basophil, or neutrophil. 33% chance. How many say neutrophil? How many say basophil? How many say eosinophil? Okay, those of you who raised hands, you got them right. This is a neutrophil, but it's a tricky one because it's got these dark inclusions in there as well. Okay, it's a little darker than you normally see. So this is actually a diseased neutrophil. So that's why this is a tricky one. Okay, now how about this cell here? Is it a granulocyte or not? It's not? Okay, then you have two choices. Is it monocyte or is it a lymphocyte? How many say mono? Okay, and how many say lymph? Okay, it's actually mono. Remember, lymphocytes have round nuclei. Monocytes have irregular nucleus like that one, and it's huge. Okay, on this side is a segmented neutrophil on the right side. Okay, this thing is fine. Oh, this one is finally dead. Okay, and these cells, what do you call it? Target cells, they're good. Okay, some of these target cells may have funny looking shape to it, like that one or even that one. But they're all target cells associated with sickle cell disease, thalassemia, etc. And these things are these red cells that have funny things like they have freckled faces associated with eating too many chocolates from high lead content, it's called basophilic stippling. Basophilic stippling. Okay, that's it. Any questions?
I think
they're just fragments. So red cells, if they're damaged, you can have little pieces of red cells floating around. It could be, like fragments of glasses or something. Any other questions? Okay.
Ask questions now, because he's probably more knowledgeable than I am.